Hello, everyone. Welcome to Science in the Age of COVID-19 seminar series. It is our pleasure to host Dr. Neville Sanjana today at our talk series. Dr. Sanjana is a faculty member at the Department of Neuroscience and Physiology at NYU's Grossman School of Medicine. Dr. Sanjana is a geneticist and employs cutting-edge gene editing technology and functional genomic screens to understand the inner workings of the human genome. In the wake of the pandemic, like many other labs, Dr. Sanjana switched gears and used his expertise to study the coronavirus. Using a CRISPR-based loss of function screen, Dr. Sanjana and his team have identified the host elements, the host factors responsible for the virus infection. We'll hear about that and some other interesting aspects of the coronavirus infection in his talk today. Dr. Sanjana, thank you and welcome. We look forward to hearing from you.
Yeah, thanks so much, Shraddha and Lauren, for the invitation. It's really a pleasure to get to present some of the recent work from our lab on COVID-19 and SARS-CoV-2. And yeah, so as you mentioned, my lab is both at the New York Genome Center and also at NYU in biology and NYU Med School. And you know, my lab is not really a virology lab. It's not a lab that really focuses on coronaviruses, but like many other labs, when we saw what happened with the pandemic, and especially being in New York City, seeing the effects here, the very early effects here in New York City, we really started to think about, what could we do as a lab? What part of our kind of unique science skill set could we contribute to helping with the pandemic? And so today, here's kind of the roadmap of what I want to tell you about. It's really two different projects, but that are heavily interconnected. In fact, one of them was almost a project that came about as an accident through working on the other project. And that's the one I'm going to start with first. So first, I'm going to tell you about a mutation that appeared in the spike protein and that kind of quickly underwent a selective sweep in our efforts to characterize the spike mutation. Of course, spike is the thing that gives the coronavirus the name corona. It's the spiky crown. And then I'm going to tell you about some of the implications from that work for vaccines and vaccine development. And then shift gears a little bit from vaccines more to kind of basic biology and therapeutics with the work that you had mentioned in the introduction, this genome-wide CRISPR screen to identify host genes. So that's kind of the plan for today. So before I jump into actually all the data, I just want to give some credit to the folks who did the work. So first and foremost, both of these projects that I'm going to tell you about today, which are available online with this bioRxiv preprint for the first part, and this recently published paper for the second part, both were led by a very talented postdoc in my group, Jarko Danilowski. And in addition to Jarko, both of them were a collaboration with Ben Tenuver's lab, which if you've been attending this seminar, I think he was the one who spoke last week, so you're probably well up to date with him, and in particular in his group, several folks helped from the Tenuver lab. Tristan was really a postdoc in his group was really the prime driver behind many of these projects, along with Jarko and my group. So in late January, early February, folks who were sequencing the coronavirus SARS-CoV-2, which causes COVID-19, started to notice a particular mutation becoming prevalent, this D614G mutation. And so you can see it here in time. You see this transition between the D variant to the G variant. And this is just from a nice review article in Science. And you can see that this transition that started around January, February really started to accelerate over the next few months, where this mutation became dominant. And really around here in late March and April, we started to think about, hey, what's going on with this mutation, and to try to understand it and characterize it. One of the first things that was very interesting about the mutation that really caught our eye was the study done by Tim Cardozo, a colleague of mine at NYU Med, who observed that the prevalence of this mutation seems to correlate if you look at it over different countries, so each dot here is a different country, and this was data from March, April, May, that this really correlates with the fatality rate, the case fatality rate of COVID-19 in that country, meaning that countries that have more of this G mutant, this new mutation, seem to have a higher fatality rate. The other piece of patient-based data that caught our eye was data from two different groups where they had done both sequencing and a qPCR test. So this is the very standard COVID test that we're all familiar with, this nasopharyngeal swab that kind of samples your brain a little bit, and then is subject to qPCR, which amplifies the viral RNA. And these two different groups dealing with two different patient populations in two different countries. So this is the University of Sheffield in the UK and the University of Washington in Seattle. They found, even though you can see that there's an overall difference in the detection of COVID-19, you can see that there is a very consistent difference between the D and the G variant. And if you just look at the fold change in this qPCR test, you can see there's about a threefold increase in detection. So lower CT values, as many of you have learned, means higher viral loads, so that there's a higher viral load in patients with the G variant. So this suggests that there's both something about the G variant being causing higher fatalities, and that even within a single individual, that if they carry this G variant of the spike protein, they have a higher viral load. And this is consistent across both the University of Sheffield and the University of Washington, with about a threefold higher load of detectable virus. So as I mentioned, this really kind of piqued our interest in March and April. And so for many of the projects in the lab, we actually use lentivirus for gene delivery into human cells. And so here what we thought is, okay, instead of using our standard lentivirus, why don't we just take the lentiviral virion and code it with the spike protein from SARS-CoV-2. And we used here either the original D variant or the mutation, the G614 variant. And inside the virus, we have a gene that carries green fluorescent protein, very well known to folks here at Janelia, where there's tons of work on fluorescent protein development. This is using a very standard fluorescent protein, GFP. And we also had a third kind of virus here where we didn't pseudotype it with really any kind of coat protein. And we transduced a few different human cell types. We transduced these lung A549 cells, the Hus 7.5 cells from that are human liver cells, and cacao2, which are cells from the colons, because we had some idea at that time that SARS-CoV-2 could actually infect multiple cell types, which we now know it infects many, many different kinds of cell types. So we tried these three viruses, these three different kinds of coat proteins, and looked to see how many of these got into cells and were able to deliver their GFP cargo, turning the cells green, fluorescent green, with this jellyfish protein. And so what we observed here is that when we didn't pseudotype the virus at all, so this is the no pseudotype condition, you can see there's really no entry into these A549 human lung cells. But when you pseudotype it with the spike, you can see that there's an increase in GFP. But if you pseudotype it with the mutant spike, you can see there's really almost an order of magnitude increase in GFP found in these cells. And so depending on the cell line, and this was very consistent whether we used cells where we had to overexpress ACE2, which is the entry receptor for the spike protein, or cells that naturally express ACE2, we see this very consistent 2 to 8-fold increase in infection of these human cells over liver, lung, colon cells with this spike variant. And just as a small aside, we had this nice little control where we measured to make sure that there was no difference in the viral prep. There was the same number of viral virions in each prep, and we made sure that this was the same between D and G. So there's not a difference in the number of viruses. It really is an effect of the coat protein itself. So, okay, so that's the observation. The natural question is why? How does D614G make these virions more infectious? And so for that, we started to really look at the domain structure of the spike protein. And what we noticed is, even though this was not in kind of a well-studied domain where this mutation is, so again, this is the beginning of the protein, the N terminus, this is the end of the protein, the C terminus, but you can see that kind of the closest well-defined domain to this D614 gene mutation is the receptor binding domain, a very well-studied domain that binds that ACE2 receptor. And so we wondered, is there some interaction here with the receptor binding domain? Maybe that's the mechanism. And this is just data from another group that was published very early on in the pandemic that shows how important ACE2 is. So here you can see the same human cells, but the difference is the ones on the top have actually have exogenous ACE2 delivered to them. So they express a lot of this ACE2, which is green in this picture. And you can see only those cells can get infected with the SARS-CoV-2 virus. So this is a staining, an immunostaining for the N protein, the nucleocapsid protein of the virus. So if you don't have strong ACE2, you can't really get the virus in. So it really does depend crucially on ACE2. And so we wondered whether maybe it just improves, this mutation maybe improves binding to ACE2 and makes this happen much more easily. And for this, we turned to some colleagues at the medical school, Jira Baba, who has a structural biology lab there, and specifically Juliana, who's a graduate student in her lab. And there they introduced us to this technique, biolayer interferometry, where we can actually take purified ACE2 and bind it to essentially a very sensitive microscope. And then we can flow over on that purified ACE2, either the mutant or the wild type spike, and we can measure the interaction by doing this kind of very high resolution microscopy. How well does spike bind ACE2? And the question is, does the mutation in spike that we see increases infection, does it somehow bind ACE2 more tightly? And though this would have been kind of a very convenient hypothesis, when we ran this result, the results were, when we ran this experiment, the results were very, very clear. That there really isn't any significant difference in binding between the wild type and the D614 gene mutant. Both of them have high nanomolar affinity here for binding. And so this doesn't seem to be the reason why the virus with the mutation is more infectious. So, okay, we had a good scientist go back to the drawing board, think of a new hypothesis. So another thing that's well known about the spike protein from earlier studies of SARS-1 and MERS, other coronaviruses, is that the spike protein is proteolytically cleaved, and that this cleavage is important, or two cleavage events is actually important to reveal what's called the fusion polypeptide, which is a set of very hydrophobic residues that make it easy for spike to stick into the target, the membrane of the target cell. And this is important for spike really being able to stick in that membrane and then deliver the viral payload into the cell and infect the cell. And so what we did is we took these two spikes and we overexpressed them in human cells. And one thing we saw very clearly, here I'm just showing one example, but we replicated it several times independently, is you can see a difference in cleavage of spike into these different fragments. So here we have a tag on the C terminus, and we're reading out the S2 and S2 prime fragments. And you can see that there's a decrease in this cleavage of spike, and this might be one mechanism that might lead to this increased infectivity. And to really understand this, I'm going to walk you a little bit through the dynamics of spike binding and entry, so you can appreciate why we think this decrease, this several-fold decrease in cleavage might be actually important. So virions, like SARS-CoV-2, are actually decorated with a mix of full-length spike, which is the S1 plus S2 complex here in purple, and ones that just have this S2, where the S1 has already been removed or has just fallen off. And if we zoom in on one of these S1, S2 full-length spike complexes, you can see that the receptor binding domain, the part that binds ACE2, is actually within this S1 subunit. It's here, it's exposed, it's on the surface, it's right where it should be to be able to bind this target receptor. And it's critically important to have the binding to S1. It helps the virus really anchor and dock at the cell. And then if you have cells that are missing ACE2, or if you just block ACE2 using an antibody, you can see that the ability of this virus, SARS-CoV-2, to infect is really much, much poorer if ACE2 is blocked with an antibody. So we've heard about antibodies being used as therapeutics, neutralizing antibodies for the virus. This is on the other end. This is blocking the ACE2 receptor. And from our own data, we are also able to observe that when we don't express ACE2 in cells, it really makes it virtually impossible for the virus to get into these cells. And the reason why this is the case has been kind of elucidated through some nice structural studies, that this receptor binding domain can be in a few different conformations, and occasionally it flips to this open conformation, which allows it to bind with ACE2. And this is only possible if you have this full-length protein. And once it makes this tight binding with ACE2, then this S1 piece can fall off, and the S2 fragment that's by itself here has this, the end has this, this fusion polypeptide, which can stick into the membrane. And so if you don't have S1, if we, as we've seen with the D614 variant, which has this increased cleavage, then maybe you don't have as many of these spikes that are able to bind. So this is, this to us seems like the most likely explanation for why we see this increased infectivity of the D614G, but certainly there needs to be more mechanistic studies, and this is really just a start. So one other thing about the study that's important to remark is that, you know, we did this all with this kind of pseudovirus system where we take a lentivirus, a gene delivery vector that we know really well in the lab, and then we put on the SARS-CoV-2 spike protein. But, you know, maybe this increased infectivity that we see in our hands, maybe that's just an artifact of this kind of strange system. You know, does this happen really with the real deal, with the real SARS-CoV-2 virus? And of course, the best way to get at that would just be to get a natural isolate, you know, something from a patient where it has this mutation. Shouldn't be too difficult because, as I mentioned, this mutation is kind of taken over compared to the D variant, which is what most people use experimentally, because that was what was in the early patients from January, February. The problem with natural isolates is that there's some linkage here. So basically, at the same time that this spike mutation arose, a protein coding variant in another gene called ORF1b also came about at the same time. And so because of this linkage between these two mutations, if you use a natural isolate and you see a difference, it's hard to really cleanly ascribe it just to the change in spike, because there's also a change in another protein. And so what we'd really prefer here is a truly isogenic model, something where the only change between the two viruses is just the mutation in spike. Okay, so another way that we can really get at this and really find an isogenic model is to take advantage of this ACE2 requirement for entry. And this is exactly what we did with help from Tristan and Ben. So here we introduced this way of complementing the D614G SARS-CoV-2 virus, the original D614 SARS-CoV-2 virus, so the original Wuhan strain full-on virus. We complemented in trans with a high amount of spike that either carries the mutation we want or is just wild type. And what the way that we're able to do this is, remember I said that most human cells are very poorly infected by SARS-CoV-2 because most human cells don't express ACE at very, very high levels. So here what we do is we co-deliver two different plasmids, one that overexpresses a lot of ACE, making it easy for the cell to get infected, and another one that expresses either the D or the G variant. And what happens then is we can deliver the virus at a very, very low amount of viral copies, such that most of the virus will be produced by these cells and then then carry either the D or the G variant, depending on which one we deliver here. And after 12 to 24 hours, this viral replication, you have most of the variants in the pool are going to carry the spike that we've fed in, because it's being produced at very, very high levels. And then we can do some microscopy to look to see whether we can see any differences in infection. And this is exactly what this data looks like. So here's 3 different time points. And what we're doing is we're staining in, so blue is all cells here. You can see there's a lot of cells, but the red dots are cells that have the nucleocapsid stained positive for the SARS-CoV-2 nucleocapsid protein. So this is cells that are infected are in red. And what you can see is basically at all time points, though it gets more dramatic at later time points, you can see that there's greater infection in the cells where the protein that was delivered in trans was the G variant versus the D variant. The G variant always has higher infection here. And this is basically all the data we collected. So we used two different initial applications of virus, two different multiplicities of infection, or MOI, and we looked at 3 different time points. And basically across every time point and every amount of virus we used, we always saw greater infectivity with the G variant, the one carrying the mutation, with this real SARS-CoV-2 full-on replicating virus. Okay, so this is a nice set of data. What can we take away from this? How does this, what do we really care about is how does this affect antibody vaccine responses? What does it mean, if an antibody or a vaccine has been designed for one variant, is it going to work with the other variant? This is, kind of the news you can use part of this. As all of us know and are following eagerly, there's many different vaccines that are in different stages of approval, and some of them getting very close to emergency use authorization. I think in the UK, it's already there. In the US, it's almost there. And the question that we really had is, does immunity to D614 spike also protect well for this variant, for the G614 spike? And so we did some very basic, I would say, computational predictions around this 614 site, which is labeled here as X. And one thing that we were able to observe is that for some of the very prevalent HLA-A and HLA-B and other HLA alleles, here I'm only showing you HLA-A to give you a sense of the data, we do see large changes in immunogenicity with the G variant. This is a computational prediction. What about, do we have any experimental data to complement it? So we, of course, don't have any patient data, but other groups have certainly done this, where they've looked at convalescent sera from people who've recovered from infection and that were originally infected with the virus that has the D variant, the original variant. And they look to see in this particular preprint, this group looked to see, how often does this sera contain antibodies that are also neutralizing against the G variant if we tested with both? And the good news is, in the majority of cases, convalescent sera from COVID-19 patients who had the original D variant could actually neutralize both D and G. But in some minority of patients, like 3 out of 41, and here's showing you what these 3 patients look like, you can see that there's a large difference in neutralization. It's worse with the G variant, which makes sense because these folks, their antibodies seem to be specific. But in many cases, we actually have antibodies that work against both. And I think there's real cause for optimism, because one thing to say about vaccines like the Pfizer-BioNTech one is they don't just use a small piece of spike. They actually use the full-length spike mRNA. So it's not, we're talking about a mutation in one specific part of the protein, but your immune system is able to generate a repertoire of responses against the entire protein. And I think one thing that's very important is, if you look at what BioNTech's preclinical data is that they've actually tested this. They've looked at vaccine-induced antibodies and tested it against 20 different mutations of SARS-CoV-2. So it's very likely that the D614G mutation has explicitly been tested by these folks. And I think this is some cause for optimism. So, that's not the only cause for optimism. Now we have at least, I would say, 3 reasons to be optimistic, which is not just this first vaccine that we heard about several weeks ago now, Pfizer-BioNTech, but there's also another mRNA vaccine from Moderna and another vaccine from Oxford AstraZeneca, all of which seem to have above 90% efficacy. And so, I think all of us have been happy to hear this news. But even with a vaccine, it's still going to be the issue that we're going to have a lot of people for the foreseeable future who are getting sick from COVID-19. And it would be wonderful if we had more therapeutic options than we do right now. And so that was part of the motivation for the work that we did to really start this genome-wide CRISPR screen, something that that we've developed and that we have a lot of expertise in, and that we've applied to other areas like cancer biology, immunotherapy, diseases like sickle cell anemia. So we've applied it really in many other places, but certainly not applied it to pandemic coronaviruses. So this was a new idea for us. But the goals of this project were really threefold. So one was to understand key host genes. So a virus is not a free living organism, right? It depends on a host. It's not really alive by itself. It needs the host to survive. So what are the key host genes that are important for the virus? And can we, when we find those genes, can we translate those genetic findings into actionable therapeutics? And when we look at these genes together, you know, is there something, are there some common pathways and common mechanisms that underlie resistance to the virus? So these were the large goals that we had in mind when we started this project. And just as the briefest of introduction, because I think for this audience at Janelia, you folks are probably very familiar with CRISPR, which really is cut, copy, and paste for DNA. But I wanted to tell you about a very specific way in which we use CRISPR in this study. And it's worth just reflecting and saying that the reason why CRISPR has been such a breakthrough, recognized with the Nobel Prize in Chemistry earlier this year, is that editing human genomes and editing many genomes has really been kind of an unreachable goal for most of genetics and the history of genetics and biology. But in the last decade, really, CRISPR and CRISPR-based tools have, I wouldn't say made it quite as easy as cut, copy, and paste in Microsoft Word, but have been kind of a quantum leap toward that goal of very easy gene editing for all sorts of therapeutics and basic research needs. So, you know, one way that I like to explain CRISPR screens, because instead of looking at a bunch of, you know, A's, T's, C's, and G's, the bases of DNA, let's think of each gene in the genome as one word in a book. So, you know, let's take, here I've got the example is from On the Origin of Species from Charles Darwin. So here's the very first sentence of this book. When on board the HMS Beagle, I was struck with certain facts in the distribution of the peoples of South America. And then he goes on, it's a very long sentence. I only have part of it here. So this cut, copy, and paste, this programmable set of scissors, which is what CRISPR is, lets you zero in because you can program it on a certain word and be able to pop it out. This is fundamentally what these CRISPR DNA scissors do. They let you really go in there, cut something out. So the way that we use CRISPR here is instead of targeting just one word at a time or one gene at a time, we scale this up. We take advantage of the fact that CRISPR is really, really programmable. And instead of targeting one word at a time, the idea here is we target all of the 20,000 genes in the genome in parallel. This is the really key idea with CRISPR screens. And then we let something about the disease we're studying or the phenotype or the trait that we want to know more about, we impose some selective pressure in this large space of genetic hypotheses, 20,000 different genes, 20,000 different hypotheses, to let us focus and say, okay, of all these hypotheses, this is the correct one. And so this is kind of what a CRISPR screen is. It lets us take a very large space of genetic hypotheses and zero in on the couple genes that are most relevant to the disease or the trait we want to study. At a conceptual level, if you understand this, you understand CRISPR screens. And so we took advantage of a library that we designed several years ago, this Gecko V2 library. Gecko stands for Genome Scale CRISPR Knockout. And so here what we have is we have 6 CRISPRs for every gene in the genome. So 120,000 CRISPRs total because there's 20,000 genes in the genome. And what we do is we take human lung cells and each cell gets a different CRISPR knockout, a different CRISPR perturbation. And then we grow those cells for some number of days, make sure CRISPR knocks out the gene, and by waiting a couple days, because when you knock out DNA, you might still have some protein left around. So we wait 1 to 2 weeks to make sure that the protein then disappears too after the gene has been knocked out. We then infect with SARS-CoV-2 at a very low multiplicity of infection, so a low amount, but still enough that would kill most of these cells. And then we let it replicate, we let it kind of do its thing, and so it is able to kill most of the cells. But what we're very interested in is which cells survive and proliferate even after being exposed to SARS-CoV-2. And we want to know, for instance, this yellow CRISPR construct, what gene does it target? That's fundamentally the question we're asking, that there might be particular genes that when you knock them out using CRISPR, they enable the cells to be resistant to SARS-CoV-2. And that's an open question. We didn't know going into this, what kinds of genes there'd be. We, of course, knew about ACE2. We know that's very important, so we'd expect that to be on the list, but we really didn't know actually too much more beyond that. And this is kind of what this data looks like. So the data comes back to us as almost a ranked list where we can start to see which genes are the most important genes for that when you lose them, you're immune to SARS-CoV-2 infection. And this is good for us, but kind of hard to comprehend. So here I'm just going to sample this list. So again, the list is 20,000 genes. So all I'm going to show you right now are about the top 50 genes. So these are some of these top 50 genes. And one thing you can instantly notice is that these genes have very similar names. And that tells us that they likely cluster into similar pathways. So one thing to notice is ACE2. So ACE2 was the one positive control we had going into this. And ACE2 is actually ranked number 8 out of 20,000. So that's amazing to see it so high up on this list of ranked genes. And if you look, you can see that many of the genes have this kind of ATP name. And so these genes are all part of this vacuolar ATPase complex. So we have really almost every component of this complex that's involved in transporting protons and acidifying these endosomes and lysosomes is picked up here. Almost every element of this retromer complex, which is important for endosome recycling, and almost every element of this commander complex is also picked up here. And so this is really great to see that these are not just random genes that are ranked near the top of the list, but it looks like we have a lot of genes like these ARP2/3 actin genes here, that a lot of genes that function together maybe as a shared pathway or shared complex. Another thing that we were very interested in when we first saw this list of genes is, well, are these genes very specific? Are they expressed only maybe in the lung or in the nasal cavity, or are they widely expressed over many tissues? And so for this, we use this beautiful GTEx data set from about 10,000 RNA-seq from 10,000 individuals, 50,000 tissues. And here what you can tell is that most of these top-ranked genes are pretty ubiquitously expressed across different tissues. You see them in many different tissues. And there are exceptions to this, like ACE2 has strong expression only in a few tissues and weaker expression in other places. But most of these genes are expressed in many tissues, which actually gives us hope that these could be interesting therapeutic targets. Another data set that had recently been published was from Neven Krogan's lab at UCSF, where they had actually overexpressed viral proteins and then perform in human cells and then perform proteomics to see what human proteins do specific viral proteins interact with. And even though the hits in the CRISPR screen don't necessarily have to have direct interactions with viral proteins, it's something that when we saw it was very, very reassuring. So here I'm plotting for every single one of these viral proteins, the top ranked hit from the CRISPR screen and how enriched it was. So you can see that some of these genes were really highly enriched in the population of cells that survived SARS-CoV-2 after receiving that particular gene knockout. And so this says that maybe some of these genes actually that we found in the CRISPR screen, they likely directly interact with viral proteins and again are just important as part of the viral life cycle. So that was very reassuring to see. Of course, a screen is really just a starting place. It gives you a lot of ideas and hypotheses. It narrows down the hypothesis space, but you really want to see some independent validation. And so we actually attempted to validate the hits from the CRISPR screen using multiple orthogonal types of genetic perturbations. I should say genetic, transcriptomic, and protein level perturbations. So we both looked at CRISPR gene editing. We looked at RNA interference to knock down transcripts, and we also used small molecule drugs to directly inhibit proteins. And so here I'm showing you the first of those levels, which is on the DNA level, on the genetic level. So here we picked brand new CRISPRs, ones that weren't in our genome-wide library, but that are designed to target the same genes. And here we picked 30 genes from the top 200 ranked genes from the screen. And here I'm showing you 3 different CRISPR guide RNAs directed against each of these 30 genes. And you can see that in all cases, all of these genes, when we knock them out with CRISPR, we see a decreased ability for SARS-CoV-2 to infect these cells. And this is just very rare to see this kind of a strong validation. Of course, some genes have very, very strong protective effects. Other genes have much weaker effects. But this really shows you that this is not just a couple of hits that we found, but that there's really depth to this list. And this was done with this kind of microscopy assay, again, staining for this nucleocapsid protein. As I mentioned, we also wanted to look on the protein level as another orthogonal way to validate the screen, but also because this could suggest possible drugs that could be used actually to treat COVID patients. And so if you just cross-reference the top 500 hits with a database of druggable genes that's available online, you can see that not all of them, but several of these top-ranked hits actually have existing drugs that have been listed as active against the proteins that are produced from those genes. And so what we did is we sampled about 10 of these genes, and for each gene we had we tried to get as many different drugs to target those genes. And what you can see is that in about half the cases, we were able to find therapeutics, small molecule inhibitors of these genes. So again, this is kind of like mimicking CRISPR, but doing it on the level of protein, not requiring any gene editing. But we were able to find several that have an above 100-fold reduction in viral load in these cells. And something that you can compare this to is you can compare this to a very highly effective therapeutic, which is remdesivir. Remdesivir directly targets the RNA polymerase. So it targets a gene from the RNA that's from the virus that is involved in viral replication. So of course, this has a very strong effect, but we know that remdesivir also has some serious issues with toxicity. And what you might imagine as an eventual therapeutic for COVID-19 would be something where we maybe will combine different therapeutics that work in different ways. So all of these work on host genes, remdesivir works on viral genes. And this is not such a crazy idea, the idea of a combination therapeutic, because this is essentially how the battle against HIV was won. HIV can be has been transformed from really kind of a life-threatening disease to something that is easily survivable and you can live with due to the cocktail of 3 different drugs that's used to treat HIV. And so what this data suggests is that by mining these top hits from the CRISPR screen and trying to identify small molecule inhibitors that maybe are approved for some totally different indication, we could repurpose them to come up with a cocktail to treat COVID-19. And this is something we're, of course, very interested in now. I want to talk about some of the mechanisms of how these genes work. So far, I've just shown you genes, names of genes, and tried to mention that we can target them and that this could be a way to prevent SARS-CoV-2 entry. But what about trying to understand, are there some themes here with these mechanisms? So for this, we actually turned to this technique called ExciteSeq, which is something that was developed here at the New York Genome Center, collaboration between several labs, including mine and the Technology Innovation Group with Peter Smybert. And ExciteSeq is a way to couple pooled CRISPR screens with single-cell transcriptomics, so single-cell RNA sequencing. And the idea here is, could we take a set of our top hits? So we designed a new CRISPR library that just targets a couple of these top hits and deeply profile these human lung cells to understand what is the whole, when we knock out these genes, what's happening inside these cells? How is the cell state changing? Which we can look at by looking at all of the RNA inside the cell. And this is kind of what this data looks like. So here, what you're seeing here is every column in this dense matrix is a different single cell. and the CRISPR perturbation that it received is, or the guide RNA and what it, the gene it targets, gene targeted by the CRISPR, is on top, listed on top here. And we also have CRISPR non-targeting guide RNAs that shouldn't do anything, don't target anywhere in the genome. And the rows here are differentially expressed genes, in this case, genes that are upregulated. And so you can see here that for several of these different CRISPR perturbations, we can see distinct clusters of genes that are upregulated. And for some of them that are kind of similar, like these two vacuolar ATPase subunits, 6AP1 and 6V1A, you can see actually a kind of similar set of differentially expressed genes, which is what we might expect. But looking at just the gene expression, because so many genes are changed, it's a little bit difficult to make out precise differences. And so here we used gene set analysis to actually collapse these to understand this at a pathway level. And when you look at it at a pathway level, the thing that sticks out most clearly is that several of these genes, in particular these 6 genes here on the right side, all result in an upregulation of the cholesterol biosynthesis pathway and several related pathways to cholesterol biosynthesis. This is something that we didn't know very much about, but we realized that this could be some sort of shared mechanism between these different CRISPR knockouts. And so what we did is we reintroduced guide RNAs that knock out these genes into these human lung cells, and we just measured total cellular cholesterol. And what you can see is in basically all of these cases with all of these, different genes here, you can see an increase in cholesterol after you upon the CRISPR perturbation. So this basically backs up directly what was suggested by the RNA, the single cell RNA-seq data. So seeing this, we thought, okay, can we use this to do some drug repurposing? And here in the literature, it's well known that amlodipine, which is a FDA-approved drug that's been approved for more than 30 years, commonly taken heart medication, it's known that in cells that it can increase intracellular cholesterol. And indeed, when we treat our A549, our human lung cells with amlodipine, you can see this very clear boost in cholesterol, quite similar to this change that we see with the CRISPR knockouts. And in addition to that, you can see, when you treat these cells with amlodipine, it drastically reduces viral infection. And we looked at this in a few different ways, using qPCR for the viral RNA, using a plaque-based assay, and also doing RNA sequencing and mapping reads back to the viral genome. And you can see that there's an order of magnitude decrease in the amlodipine-treated cells versus those treated with a vehicle control, just DMSO. Another kind of more mechanistic question. So that was one interesting mechanism that we discovered with this cholesterol regulation. Another interesting mechanistic question that we had, because it was something we knew about SARS-CoV-2 entry, is do any of these top-ranked genes impact the cell surface expression of ACE2? I showed you in the earlier part of this talk that ACE2 is absolutely essential for having entry of SARS-CoV-2 into human cells. And so we wondered, maybe some of these genes that are not ACE2, maybe they work by somehow preventing ACE2 from being available for binding. And indeed, we were able to find some genes that do this. So here's one of these genes, RAB7A, and you can see what we're doing here is just labeling the cell surface ACE2 with a fluorescent dye and then doing flow cytometry. And you can see that compared to a non-targeting guide RNA, this NTG1, if we target this gene RAB7A with either one CRISPR guide or another CRISPR guide RNA, you can see quite a shift in a decrease in ACE2 levels. And so this kind of data, I've just summarized it for many different genes from our top hits from our CRISPR screen, and we were able to identify 2 different genes that seem to have a significant effect on ACE2 availability on the cell surface. So one natural question you might have is, okay, if it's not on the cell surface, where did it go, right? We're not knocking out ACE2, so ACE2 still exists, we think. So where does ACE2 end up? And here we did some confocal microscopy here. So here you can see ACE2 is normally here on the cell surface. You can hopefully see these faint outlines here on these cells. And here's the case of the RAB7 knockouts. And here the story is really different. You can see that the ACE2, instead of being along the cell surface, is getting stuck inside the cells. And you can also quantify this right here. So our question is, where is this getting stuck? So we co-labeled ACE2 along with some markers of either early endosomes like EEA1 or late endosomes and lysosomes like lysotracker. And you can see this really nice alignment between ACE2 and EEA1. They overlap tightly, and there's much less alignment of lysotracker. So it looks that ACE2 is really getting stuck or sequestered in endosomal compartments in these RAB7A knockout cells. And this is not one thing that we wondered was whether this might just be an artifact of the fact that in these particular lung cells, we actually overexpress ACE2 to make it easier for SARS-CoV-2 to infect. So we also tried this in two different cell lines, a lung cell line and a colon cell line, that naturally actually express higher levels of ACE2. So there's no ACE2 overexpression. And in both of these cases, when we knock out RAB7A, we see a really similar decrease in the amount of ACE2. So this really seems to hold true regardless of what human cell type we use. So how are we doing here? Oh, good, perfect. So just to summarize, in the first half of the talk, I told you a little bit about our work to characterize the D614G mutation. And we were very interested to understand its mechanism. We measured if there was a change in affinity for Spike's receptor ACE2, and we didn't see any change in receptor affinity, but we did see a difference in the cleavage, the proteolytic cleavage of Spike. And this might be a potential mechanism of why this is more infectious, and more infectious not just with the pseudovirus, but also with the full SARS-CoV-2 virus. And I presented this transcomplementation assay that allows us to really measure this mutation. And in the future, any other mutation that might occur in the spike protein. This allows a really rapid way to do this. The second part of the talk, I told you about this genome-wide CRISPR screen where we knocked out every gene in the genome, and we looked to find genes and mechanisms of resistance to SARS-CoV-2, and made some very initial attempts to try and take the results of this CRISPR screen and translate them all the way into therapeutics that hopefully one day we can use as a treatment, either as a monotherapy or perhaps as a cocktail of multiple therapies together to take this, to have hopefully a similar success story as to what we've seen over the last 3 decades with HIV. So with that, I want to thank the people from my lab and the many collaborators that we have. I especially want to just mention Jarko, who really led all this work in my lab, and also mention Tristan and Ben, who were part of both of these projects that I told you about today. And I think that's it.
Awesome. Thank you, Neville. That was pretty amazing complement of things you did. So remind people, either go ahead and type your question into the Q&A box, or raise your hand in the participants menu, so it makes it easier for us to call on you. Neville, I'll go ahead and start out. Something that really jumped out to me from the GTEx expression atlas, ACE2 is really barely expressed in the lung. It's 2 orders of magnitude lower than other tissues. Why do you think the virus goes for this thing?
Yeah, I think that's a really great question. So, to be very honest, I don't think our work really speaks to this. I know there are other groups that have identified some other possible entry receptors, like I think one of them is neuropilin. Mm-hmm. And so, I think it's a combination of things. So one, I think there's likely some alternative entry receptors, but it also might be the case that relative to expression of these other genes that you actually don't need that much ACE2. So even very low levels of ACE2, which is probably the case for most of these tissues, that they express very low levels, that probably is sufficient for entry. And for people who study, like neuroscientists who study receptors, the expression of them is typically not very high. relative to like a housekeeping gene or something else, or intracellular enzyme.
Yeah, okay, Brian, go ahead and raise your hand, and Sarada will turn you on to ask your question.
Brian, unmute yourself, please. You can ask your question.
Hi, can you hear me okay?
Yes.
Yeah, perfect.
Okay, I'm just wondering about the generalizability of your modifiers of SARS-CoV-2, CPE, and especially if you've looked at any cell lines that have endogenous levels of ACE2 expression rather than heterologous expression, and if you have a sense of whether or not the same hits would turn up.
Yes, actually, I don't have that. I have this, but I don't have that exact slide. So in one of our supplementary figures, so there's a few things to say. So the first thing is that we did the screen in an ACE2 overexpressing cell line. So it's certainly you might have some differences in other cell lines too. And that's, there's actually several different both published work and preprints with CRISPR screens in different cell types. And that's been, one of the amazing things about COVID is that there's never just one paper. There's many different papers, and some of them did it in pretty much our exact same system, but used a different CRISPR library and found the same hits, which I think is really great. Love seeing that kind of reproducible science. But there are other groups that did in other cell lines, some that overexpress ACE2, some that don't. And I think there's going to be very interesting differences there. Speaking to our data, this is something that one of the reviewers of our paper asked us to do. And so, of course, we did it. And so for the top hits in our screen, we actually did it. We also looked in a different ACE2 overexpressing cell line, which was the HUS 7.5 ones that overexpress ACE2. We were able to validate it there. And then we took a selection of those hits and did it in another cell line that does not overexpress ACE2. And so to the degree that we've tested it, which is certainly not, the same all 30 hits from the initial validation that we did in A549, but the degree that we've tested, we find that most of the hits really do generalize. I expect some of them are going to be cell type specific, but many of these seem to be tied to very basic processes. A lot are involved in endocytosis, in things that you wouldn't actually expect to be very cell type specific.
Sarada, you have one now?
Yes. So, Neville, a very elegant study to delineate the mechanism of infection and identify possible therapeutics. So, a question to you is, have you done or are you planning to do similar studies for other members of the coronavirus family, especially the SARS one and the MERS, to see if, how, because, these infections are similar in certain ways, yet they're different?
Yeah, that's a great question. Yeah. So do we plan to do CRISPR screens in other related coronaviruses? We don't have any plans to do that, but there's another paper that was published basically back to back with ours a few days earlier in Cell. It was published online. This is from Craig Willen and John Dench. And their study actually, it's not done in human cells, it's done in the Vero cell line, which has commonly been used for a lot of the COVID-19 biology. And there they actually do look at a few different coronaviruses and make some comparisons there. So I think if for folks who are interested in that, that is definitely the paper to check out. In our study, we do some comparisons, not like exactly what you mentioned, but we actually look at other CRISPR screens done for Zika and pandemic flu, H1N1 flu, and we really weren't sure what we'd see, but actually surprisingly, there is some substantial fraction, of overlap between all three of those screens and, pairs of screens too, which is interesting. It suggests that some of the hits that we're getting are really some sort of common viral mechanisms that many different viruses, even viruses that are very distantly related, are all using together.
Interesting. So if you don't mind me asking, what families are those that seem to be similar between these infections? Are they
what kinds of proteins are similar between?
Yeah, or is there any certain specific?
Yeah, I'm trying to remember exactly what those are. I wouldn't want to misspeak, so I have to look back at the supplement. But it's one of our supplementary tables basically shows the overlap between all of those three that we compared. But we were very lucky because, for instance, The Zika screen, I think, was done last year. The influenza screen was done earlier this year. So this is all really kind of recent data. And it would be, I think, from a drug discovery and development, we just did this because we thought it would be kind of neat to compare, and we weren't expecting anything and maybe no overlap. But seeing the overlap was nice to see. And I think from a drug development perspective, there might be some deeper insights that we can get by looking at these dependencies that are kind of multivirus dependencies. And maybe there's things that could be shared between all of these. And that's not that crazy because, drugs like remdesivir have been used to treat other viruses, completely different viruses. And so, they weren't designed for SARS-CoV-2. So this is not, I guess now in retrospect, it's not that, it shouldn't be that unexpected what we found.
Exactly, because similarity between flu got me thinking about repurposing. Yeah,
no, it's something, we're, as I started to talk by telling you, we're not virologists, we're also not drug development people. And so, that I think is something that somebody who's got that interest and that expertise, that's probably a beautiful data set to mine further.
Neville, but piggybacking off that, can you talk a bit more about the drug hits that came out of your screen? Like, how much do they overlap with what other people have found with repurposing? What should maybe be in the quiver?
Yeah, so that's a great question. So how much? Yeah, so the other approaches that have been taken, for instance, there's a large consortium of folks supported by DARPA who have undergone, I would say, really traditional but ultra high throughput, amazingly high throughput compound library screens. So there, instead of, we started with a genome-wide CRISPR library screen. So these folks took a different approach, is that they didn't really care about the genetics. The question, because the question there is really, let's just get to the drugs as quickly as possible. And I think that has its merits for sure. And what they did is they just took libraries of thousands, tens of thousands of compounds and screened them in a very similar way, human A549 lung cells, this kind of thing. And I would say largely that the compounds that have been discovered there, so the hit rate there tends to be very low, and it's much more labor intensive because it's you have to do these kind of one at a time. The cool thing about the CRISPR pooled screen is one person can do this in a couple of days. It's a pooled screen. You treat it like just a single reagent. That's one really nice advantage. So you can do this, without a consortium as a single lab. But so the hits that I've seen that have come out of those have actually been quite different than what we found with the CRISPR screen, and that's not very surprising because here, the way that we get to a small molecule therapy is we find a gene and then we look for inhibitors of that gene. So we're specifically looking for, in some ways, very on-target effects. And drugs are quite famous for having lots of interesting off-target effects. And that's a really nice thing about the compound library screens, is they kind of just sum over all of that. They're just looking at basically the endpoint you care about, which is blocking the virus. And so we would expect those to be different, and indeed that's the case. Other efforts, like for instance, work by Ben Tenuver's lab has taken a different approach, which is to just transcriptionally profile cells after they're infected with SARS-CoV-2. And there they were able to identify mechanisms like the cholesterol mechanism just by going through this transcriptional profiling, looking to see this interferon response, things like this. And so there is some overlap with those kinds of more genetics, genomics-y approaches.
Mm-hmm. And the flip side of that, do you know, so the drugs on your chart that drove it into overdrive, like vorinostat, the HDAC9 inhibitors. I did a quick Wikipedia study, and so apparently these are lymphoma patients. So presumably they're going to, already be sequestered and not going to, the mall.
Yeah. So that maybe I can share my screen again for a second.
Yes.
Is that possible? Let me see here. Let me get to the right part here. Okay. So let's take a look. Share my screen. Here we go. So this is, so you're looking down here and you were intrigued by this very weird piece of data. Let's see, make this big here. This weird piece of data that these guys, the pan-HDAC inhibitors, seem to have, they're increasing killing. They're making the virus kind of worse for you. Yeah.
Sure.
What's up with that? So this is also something that we were kind of put through the ringer on through the review process. You can see that we've shaded these bars, and I cut off part of this figure. So there's another part of this figure that Lauren is probably looking at that actually shows the viability of each of these drugs. So what is the impact? It's great if a drug reduces the virus, but it's not great if it also, if it kills your lung cells, then that's no good. And the good news is pretty much all of these compounds have minimal effect on viability. And we didn't even do a proper dose response curve. We just tested everything at one dose. The next step, like the obvious next step if you're a drug developer, is to do a proper dose response curve. Our goal was just to get through as many drugs as possible. We did not do that. It should be done. Now, what I can tell you is the viability for these, if you look at the viability chart, these drugs are outliers. So the reason, what we think here is the drugs induce pretty substantial toxicity. Like I think one of these, the cell survival is like 50%. And the viability measure is not done with virus. It's just the drug alone. So we're isolating the effects of the drug. So it's likely, and I think this is well shown in a lot of other literature, that if you completely inhibit all the HDACs, which are epigenetic modifiers in our genome, very important for regulating gene expression, that this is probably pretty toxic. And so I think the good thing is, even though these guys are not super effective, these two, which are more selective HDAC inhibitors, don't have the same viability issues that you see with all of these vorinostat, belinostat, pan-HDAC inhibitors. So this data I don't think is trustable right here, which is why we've shaded it like this. I don't think you can really say that it accelerates the virus, because you're actually conflating the toxicity of the drug with the viral killing. Yeah.
Yeah. I've got a question on the first part of your talk, D614G. I remember like March, April, I was starting to live on Nextstrain and GISAID, looking at all the mutations coming up. And there were a lot of mutations that were becoming much more prevalent. 614 was, it was only one of about 10 that looked like they were going through massive enrichment. And, there's lively debate on Twitter, how much of this is founder effect, how much of this is real. So my question is, now we've got these second tier mutations like A222V, Y453F. That's one of the Danish mink farm mutations. But when is one of these worth moving into the lab for very careful study? What's the threshold?
Yeah, I think that's a great question. So I think, just to answer your thing about D614G, I think there was a lot more uncertainty when we got our data, and we actually kind of held off on publishing it. We were, because again, we just thought, this is our first time here, like we're not really experienced at this. But we essentially, another group put a preprint online, and we put ours online like a day or two later because it was good. And that group was a very, 20 years studying coronaviruses and had been involved in the discovery of ACE2 actually as the receptor for SARS-1. And so we felt very confident after seeing that. But in fact, I think our confidence was affected by a lot of the folks in the months before that, in May and April, who were basically saying that this could likely be a founder effect. Now, since then, I think it's been rigorously shown by now, I've lost track of how many groups, let's say around 10, probably more than that, that D614G is probably not even if there is some founder effect that could be part of the reason why it took over so quickly, it's certainly not the only reason. There is ample experimental evidence in not just our system, but many other systems, including in vivo models, that really shows that this is a more infectious variant. to your other point, because what our lab tends to do is pooled mutagenesis screens, and that's something that's very exciting to us. I've been trying to get people in lab to think about, hey, can we really scale this up? How can we we want to be like a rapid response team, right? For the future, like essentially for this virus or any future pandemic virus, we would love to have a way to very quickly characterize these mutations. And so we've been trying to think about different ways to do that. Certainly one thing that's really great is gene synthesis technology, the same pooled synthesis that we use to actually develop the CRISPR libraries, but that's quite easy because we have to synthesize just these short guide RNAs. But the same way we make, print out 120,000 of those, that same technology is now being used to assemble larger and larger gene fragments. And so it would be wonderful to have kind of like a library of barcoded spike proteins and then put them through different sorts of in vitro and vivo selection. I think that would be the most efficient way to go about this. And this is something that we're very interested in doing in the future. We haven't done it, but I'm super interested in doing that moving forward.
Yeah, we had Jesse Bloom on who talked about show data with his every single point mutation to spike. And yeah, that's a pretty amazing data set.
Yeah, I think that's the way to do it. It's just, it's so comprehensive to have that kind of thing. And you can almost be predictive. You can say, well, if this arises, then this is going to be an issue. And so, yeah.
Oh yeah, no, I had like 3 mutations flagged from his discussion section have appeared in the wild, and people are like, oh shit, that one.
And I mean, the thing is that the single mutations maybe are doable. Then you start, you brought up the idea of combinatorial, like you have D614G and then you have another one. And I think that's, to efficiently search that space, I think is challenging, but that's, that would be, I think, a really amazing scientific leap if we could do that for sure.
Okay,
Yeah.
I'd like to add one question to that. So we've seen that the SARS coronavirus infection correlates with age and sex. So is there anything known about the infection rate of the D614G mutation? I mean, I guess my question is there, at the population level, do we know that this mutation is more infectious at different age, across the age and sex?
Yeah, that's a great question. I feel like there are folks who have looked at that. I don't know that off the top of my head. I feel like the best reference for that is a paper in Cell that appeared over the summer. I'm going to try and kind of search the memory bank here. So I believe the group that produced the University of Sheffield data that I showed from the UK, in the same paper where they present that, I think there is also some epidemiological analysis of the breakdown, because they did this kind of testing plus sequencing, and they have it for quite a large population. I think that group. The other reference for that's worth looking at, where I don't think there's an effect, is there's a beautiful GitHub kind of paper by Trevor Bedford's group that came out very early on, that shows the University of Washington qPCR data, so basically the other data set that I showed, that looked for that kind of correlation too. It's hard to say, whether it might be a power issue, but I don't think there's at least a large effect, is my right. But those, I would consult those 2 studies. I'm not an expert on those.
Thank you. I'll look that up.
Yeah, Neville, thanks so much for your time. Thanks for showing us your beautiful data. It was a real pleasure to have you.
The last time I was at Janelia, I talked about axon guidance, so it's fun to change things up.
I know how you feel. So, audience, please join us next week, same place, same time, for Michel Nussenzweig, who is a HHMI investigator and professor at the Rockefeller. So, Neville, thank you again.
Yeah, thanks so much, Lauren and Shraddha. Thanks, everybody. Bye-bye.